All right, in this video, I'll be looking at 2.4 plasma membranes, and we'll continue with the Unit 2 about the cell structure and function, generally getting a just basic idea of what plasma membranes are and how they work in this video. So first of all, the function of the plasma membrane, and just up front, I may use the word cell membrane some just because it's so programmed into my head. Cell membrane and plasma membrane for our purposes is basically the same thing. But as far as the AP exam is concerned, you will almost always see plasma membrane being used for that term or for this thing. So the function of the plasma membrane is to provide a boundary between the interior of the cell and the exterior of the cell. It's important to note that the interior and exterior of the cell in living things is going to be water. Because we are mostly composed of water, or most living things are composed of water or have water in their environments. And so when we talk about how the cell or plasma membrane organizes itself, that will be crucial. The plasma membrane also controls the flow of materials into and out of the cell. And so that's really important as well, because it's going to serve a function to basically block things from entering in or allow certain things in at certain points. Plasma membrane is made up of lots and lots of these molecules called phospholipids. Phospholipids are called amphipathic, and what amphipathic means is they have both a hydrophilic and a hydrophobic portion. The hydrophilic portion, or the water-loving portion of the phospholipid, is the head. You can see that little ball there. It has a phosphate group, which has a charge, which is going to cause water to be attracted to it, because water is a polar molecule. And so this is the polar portion of the molecule. And the tails are two fatty acid chains, which are extremely hydrophobic, and they are going to not like water. And so the way then that this molecule will form, as you kind of look here, you can see the individual phospholipids. When they are exposed to water, they will spontaneously form this bilayer, which is sometimes called a phospholipid bilayer, whereas the polar heads are pointed toward the water. You can see toward the inside and outside of the cell, and the hydrophobic tails are pointed toward each other. And so it promotes this really airtight or watertight system, so that the cell is completely separated from its environment. And so you have this extremely hydrophobic inner portion and hydrophilic inside and outside. That is going to affect the kinds of proteins that can be used and the way that proteins are used. There are two kinds of proteins that are used in cell and plasma membranes. You have peripheral proteins, as you can see there on the right. Peripheral proteins are found on the periphery of the cell or on the outer cell. They do not go all the way through the plasma membrane, but rather sit on the inside or the outside. They are typically used in enzymatic function, which we will talk more about in a later video. And then there are integral proteins, which go all the way through the membrane. And so these proteins too have to be amphipathic as well, where they have a hydrophilic portion and a hydrophobic portion, so that they can rest in the plasma membrane. The plasma membrane is going to have several functions. Move myself out of the way there. And so here's three of those functions that we'll list. There's the transport function, which speaks for itself, transports materials across the membrane. An enzymatic activity, which is going to serve as a catalyst for cellular reactions. Signal transduction activity, which is essentially cellular communication, communicating from the outside to cause something to happen on the inside. We'll have a unit about that. Cell-to-cell recognition, so how one cell recognizes another cell as being self or non-self. This would be like an immunity function. Intercellular joining, physically joining two cells together so that they don't separate. And attachment to the cytoskeleton and extracellular matrix, which we don't have to go into detail with the extracellular matrix, but I think the best way to understand this is it allows the inside of the cell, you can see the connections here, the cytoskeleton, kind of to understand what's going on the outside of the cell, and gives there some continuity between the inside and outside skeletal portions of the cell. And so next we're going to look at this idea of the fluid mosaic model. In this picture, you can see the phospholipids are kind of moving around. You can see something that looks like an egg going by, and a green egg going by too. Those are representative of what would be like proteins in the plasma membrane. And so when we say fluid mosaic model, it's a mosaic in that there are multiple kinds of things going. You have phospholipids, you have proteins, you have other going on the cellular membrane. And you also have, it's fluid, so it's constantly moving. It's not a static structure, but we say it's a dynamic structure. It's constantly moving around, constantly changing positions. I like to think of it, imagine if you were looking at a crowd from a high position and you watch the people in the crowd moving around. They're all just kind of moving around. Nobody's static. And so it's kind of the way I think about it. So that's what we mean when we say the words fluid mosaic model. One couple other things that you find in the plasma membrane. First, there's cholesterol. Cholesterol kind of is a steroid that kind of wedges itself between the phospholipids at random intervals. And the purpose of the cholesterol is to regulate the fluidity of the bilayer under different conditions. If it's really cold or if it's really warm, that can affect the fluidity of the lipid bilayer, which can affect the organism overall. And so this cholesterol allows there to be some continuity and consistency in that lipid bilayer fluidity, and also the presence of certain kinds of carbohydrates. These carbohydrates are attached to proteins or lipids, and so you see these, they're called glycolipids and glycoproteins, and their main function is just cellular recognition and communication.